Okay, so this is work by grad students Van and Samira in my lab. So Candida albicans is an opportunistic human pathogen. Um, it's a good system for the development of novel antifungal approaches. Um, there's already quite a few drugs out there, whether fungistatic or cidal. I'll foreshadow and say that this talk is primarily focused on fluconazole, but we've done analogous experiments with a set of additional compounds, different modes of action, etc. Uh, Classic drug resistance is usually phrased in terms of mutations in the gene that codes for the target of the drug. So, for example, mutations in ERG11 render fluconazole ineffective. For our purposes here, we can think of this kind of classic drug resistance, genetic resistance, as being an individual at some time point early before drug exposure that develops a latent advantageous mutation, and when exposed to a drug at time t0, that colony just does just fine, and we can say that they're resistant. We're primarily interested in this green subcolony that doesn't harbor such a latent mutation. Instead, it relies on a epigenetic response to deal with the drug challenge. It may fail, or it may succeed for a while but find no long-term solution, or it may succeed for long enough that a novel de novo genetic mutation arises, conferring long-term resistance. We're really interested in the question of whether genetic resistance could remain inherently epigenetic, meaning that there's no actual genetic event, but the cell's transcriptional response and processes that rise to the occasion of the drug challenge are sufficient for long-term survival. So we wanted to explore these different, what happens in these colonies. So to do that, we imported a nanoliter droplet sequencing approach for single-cell transcriptomics that had been optimized in mammalian systems. So we brought this in for Candida. So in a sentence, basically each individual in your colony is encapsulated in an oil droplet alongside by microfluidics. Inside that droplet is also a microparticle or bead, and the responsibility of that bead is basically to capture and barcode all of the mRNAs in the cell. I should say that it's not just mRNAs. We could also capture DNA or other objects, even proteins. And basically, once that barcoding occurs, you can use next generation sequencing and then bioinformatics to deconvolute and tell us how much of each transcript was present in each cell. Okay, so we optimized that protocol and took into consideration the cell wall, the spheroplast, and the large hyphae morphologies we had to deal with in the Candida setting. And it also required the development of novel informatics to take these complete transcriptomes and genomes from many individuals and allow us to do robust data science to make meaning of this data. So what did we do with this system? Okay, so what we did is we profiled untreated 5314 cells, C. albicans. That means the single cell sequencing, transcriptional, and the whole genome sequencing of DNA. And then we subjected them to drugs, fluconazole and others, and profiled after 48 hours, 2 days, and profiled after 72 hours. And so the real question here is then, can we find evidence in those profiles of processes we think might be active at that tolerance to resistance transition, and try to discover something new about its mechanisms. So this is what the data looks like. This is a so-called UMAP clustering. You can think of UMAP as being a fancy two-dimensional clustering, maybe like T-SNE, some of you might have heard. Each dot here represents a cell, and the color represents the sample of origin. So for example, here, this is primarily untreated cells. And you can see already that there's quite a bit of heterogeneity, and some drug-treated cells are intermixing. So here, two cells are close in the UMAP if they have similar transcriptional profiles. Otherwise, they're going to be quite far apart if they have different transcriptional profiles. Interestingly, like caspofungin and fluconazole survivors at 48 hours intermix here almost perfectly, whereas nystatin 48 survivors seem to be quite a bit of an outlier. The 72-hour later time points are interesting. Like for fluconazole, not all of them, but some of them drift back to look a little bit more like untreated cells, which might make sense. So basically, this is the entry point to single cell transcriptomics, and the goal here is to make sense of this cluster and try to withdraw biology. So one way we might do this is to take this UMAP clustering and project the expression of different pathways and processes. So for example, here I've looked at the average expression of efflux pumps. So these guys here have high expression of the efflux pumps, whereas these guys are very low. And we can look at different signatures from literature of hyphae morphologies, or we could look at any number of other stress or basic biological maintenance pathways in the organism. So we did that process for quite a while, and what we found for fluconazole, but it's really true for most drugs that we've observed, is that there's at least two responses. I don't want to anthropomorphize too much here, but we do observe the cells making a decision to go towards a response A, or aggressive, and these cells over here, when we look into their profiles, they're throwing everything back at the drug to survive. They're upgrading their efflux pumps and cell wall maintenance, and taking a harsh stance. Whereas these response B individuals, for boring, they both basically shut down their metabolism and really try to wait out the storm. You could ask, well, what causes a cell to make a decision one way or the other? And we don't know that. What we can see, for example, is that the environmental stress response from gash built in Saccharomyces has evidence of fluctuating in the untreated populations. So perhaps, for example, cells that happen to be expressing a certain level of DESR upon drug exposure go one way, whereas the others go a different way. And one could ask about the mechanisms for, underlying mechanisms for how to go from tolerance, epigenetics to genetics, and there's been a series of very nice papers about that, including an effort from the Broad Institute with our collaborators Como and Regev, where they look at 43 clinical isolates from patients, Candida, and they found hundreds of genes involved in a loss of heterozygosity and mutation polymorphisms. And their time scale was on the order of months between profiling. So that means that genetic events that might confer advantage have time to expand in the population. And so they're witnessing, with the sequencing, they're observing that selection and expansion. In our experiments, we also have the DNA profiles, but our time series are more refined. And so between drug exposure and profiling, it's only a matter of 2 or 3, maybe 6, 12 days. So there's not really time for those advantageous mutations to expand in the population. Rather, what we observe are hotspots, sites in the genome that tend to have mutation. And in fact, there appears to be lots of them, on the order of hundreds, involving many different genes. Now we've really focused in on 3 guys, HSP78, PST1, and FMO1. All 3 have properties that make them, I think, good candidates for being involved in resistance to processes. They are all highly expressed in only response A or response B, so they're different. They have differential behavior. and they persist. So no matter what drug you give, really, the mutations arise in these genes, and they stick throughout our time series up to 12 days. So yeah, now that's kind of motivated a whole bunch of studies to try and see if they're causally implicated in these in resistance in Candida. And so yeah, I leave it there, and once again thank Van and Samira for their effort here.